Hi, I'm Phil Bourne, Director of the Protein Production and Characterization Core Facility. Welcome to the tour. For the growth of bacteria in yeast cultures, we've got refrigerated shaker incubators, and when it's time to harvest the cells, we've got a centrifuge with a rotor capable of spinning down 6 litres of culture in one go. For cell lysis, we have an Emulsiflex high-pressure homogenizer capable of lysing yeast and bacterial cells quickly. For protein purification, there are 3 options available. We have the ActaPure FPLC system, capable of all types of protein chromatography, including size exclusion, ion exchange and affinity. We have the low-pressure ActaStart system that is useful for ion exchange and affinity chromatography. And finally, we have the Bio-Rad NGC system that is also capable of size exclusion, ion exchange and affinity chromatography. We have SDS page systems and Western blotting that are used to check the purity of protein samples throughout the purification process. A ChemiDoc imager is available to record results and is capable of imaging SDS page gels, Western blots and DNA gels. It also has both chemiluminescence and fluorescence detection capabilities if required. The ChemiDoc can be used to analyze both the molecular weight and quantities of protein or DNA samples in a gel. A nanodrop spectrophotometer is available for accurately measuring protein and nucleic acid concentrations from only 1 to 2 microliters of sample. Here we have the Microcal Peak ITC isothermal titration calorimeter from Malvern Panalytical. It provides detailed insight into binding energetics. This system delivers direct, label-free and in-solution measurements of binding affinity and thermodynamic parameters. The Octet Red 96 biolayer interferometer from Sartorius is a label-free, real-time analysis instrument measuring kinetics and affinity and screening protein-protein and protein-small molecule interactions. It can also be used for measuring the concentrations of proteins and other biomolecules. The PUNC dynamic light scattering instrument from Unchained Labs is used to assess homogeneity and monodispersity of a sample prior to crystallization trials. It can also be used to determine the size of polymers and nanoparticles. The Tensor 2 FTIR from Bruker is used for the analysis of conformation and structural transitions of proteins. It's particularly useful for confirming that mutant proteins are correctly folded. If you have any questions about any of the services that we offer here at the PPC Core, please do not hesitate to contact me.